Ladies and gentlemen,
Professor Jack Rutherford once said, even in this high-tech age, the low-tech plant continues to be the key to nutrition and health. However, every year, thousands of primitive plants die due to the large-scale use of oil-based synthetic fertilizers, pesticides, and higher inputs that they expose to the soil, causing soil degradation and the diminution of the soil's quality, which is worrying, concerning, and vexatious.
hello, my name is Gonson Mapaku.
And I'm Sudesh Kanu.
We are grade 11 students from Bilik International School, and we are here today to talk about the effects of early probiotic supplementation on germination of Arabidopsis thaliana.
The pressure to produce enough food intensifies as the world's population increases over time. The exponentially increasing global population is projected to surpass the scale of 9.8 billion people by 2050. With more people comes the need for more food, and since agricultural commodities are the world's main source of nutrition, it's important that agricultural output keeps pace with the ongoing population. However, various environmental stressors, including the looming threat of climate change, have put agricultural industries under immense strain. Another rich environmental stressor is the impact of soil degradation on the agricultural industries. Farming in continental food has become more and more difficult, with the slew of deleterious effects caused by this soil degradation. These detrimental outcomes include the depletion of vital nutrients and the emission of fecal bacteria and fungi that synthesize essential organic material for the plants through the soil.
This imminent future disaster calls for a change in the way we cultivate our food.
As a result, this study was conducted to investigate how probiotic bacteria like Arabidopsis,
RL,
Bacillus subtilis,
BS,
and Stenosphorescens
PF,
would impact on the growth of Arabidopsis thaliana when applied to the seeds. With regards to experience, we carried out probiotic precautions such as preparing the seeds, the bacteria, and the essential conventional equipments.
yes. Escherichia coli, Bacillus subtilis, and Stenosphorescens were stripped onto nutrient agar plates. Rhizobum leguminosum were stripped on Rhizobum X plate. We used a sterile plastic inoculation loop to pick up single colonies and spread them on their respective plates. We incubated at 25 degrees Celsius the Rhizobum leguminosum and Stenosphorescens. And we also incubated the Bacillus subtilis and the Escherichia coli at 25 degrees Celsius. We kept changing the plates in which the bacteria were placed weekly to keep a growing population of bacteria alive and prepared for when we were ready to use them. After this, we stripped each culture of bacteria onto liquid medium. Nutrient broth was used for Escherichia coli, Bacillus subtilis, and Stenosphorescens, and Rhizobum X broth was used for Rhizobum leguminosum. After using a centrifuge to spin down and separate the cells of the bacteria for 30 seconds at the speed of 6,000 RPM, we combined the cells with a phosphate buffered solution or PBS. We repeated this process once and the different solutions were used as different treatments for the Arabidopsis seeds.
The 7 experimental groups and the control groups that make up the different treatment groups are depicted in Table 1. Groups 1 to 7 are composed of different combinations and quantities of Bacillus subtilis, Stenosphorescens, and Rhizobum leguminosum. The water-only and Escherichia coli are the negative control groups.
Yes. For treatment groups containing multiple species of bacteria, we took equal measurements of each solution to create a 500 microliter mixture.
On seed treatment, we thoroughly rinsed the seeds with 500 microliter of distilled water, 5% sodium hypochlorite, and finally 70% ethanol. We then soaked the seeds for 30 minutes, and after the seeds were thoroughly sterilized, we placed them on clear square plates, which were previously filled with 0.10 agar solution On seed treatment, we thoroughly sterilized the seeds with 500 microliter of distilled water, 0.8% of agar solution, and 5% sodium hypochlorite. We then soaked the seeds for 30 minutes and were placed in clear glass plates.
Regarding germination and growth, the store plates containing the seeds were placed on a rack at 45 degrees length under a fluorescent light so that the seeds could germinate. The fluorescent light was set to be lit for a 16-hour photo period so that the seeds could develop into seedlings and germinate. The square plates containing the seeds featured 1.3 cm grid lines, which we used as scales to measure the root length of the plants. After the 2-week span, we counted and recorded the number of seeds treated, and in addition to how many of these seeds germinated. In total, we had 233 seeds treated, from which 135 germinated and 98 did not germinate. Moving on, our hypothesis was as follows. The greater the number of probiotic bacteria from multiple genera, the greater the growth of the root of the plant. To prove this, we measured the total root length of the plants on each of the treatment groups and calculated the average length per group. The total root lengths among the treatment groups are depicted in this box and in this plot. The X on each box denotes average value. We observed that apart from the group containing Bacillus subtilis and Pseudomonas fluorescens, the other treatment groups of bacteria had larger root lengths when compared to the control groups. We also counted the number of branches per plant in each treatment group. The number of root branches per plant on each treatment groups are depicted in this box and this plot, where the X on each box denotes the average value. We observed three things. First, groups containing Rhizobum leguminosarum had the highest branch counts. Second, the presence of bacteria led to higher branch counts. And third, as we increase the number of bacteria, the number of branches also increased.
However, we rejected the hypothesis that the greater number of probiotic bacteria from multiple genera would lead to an increased promotion of root growth. On the contrary, each seed behaved differently in each situation. In image A, distilled water, a plant from water only negative control group contained one root that did not diverge into any direction. On images B, C, and D, the roots were separated in a similar fashion, with small roots and branches. And finally, in image E, composed of all three genera, BS, PF, and RL, contained one main root that diverged into 7 small roots which branched out into different directions. In conclusion, the results obtained from this experiment will contribute to the knowledge needed to develop a viable alternative to the addition of fertilizers and to make agriculture more and more sustainable for the sake of both environmental and global survival. As Agmandino said,
always do your best.
Whatever you plant now,
you'll harvest later.
Thank you for your attention.